So here's a schematic of a cell, and we can highlight different parts of it in color. So let's go through some of these components just to make sure that you can identify them. So if we look at the actin cytoskeleton shown in green here, we can see that that is at the periphery of the cell, and there are cables of microfilaments just at the periphery of the cell, although there are actin microfilaments elsewhere as well in the cell. The chromosome, of course, is in the nucleus here. We're showing chromatin, and here's the nucleolus, the region where ribosomal components are manufactured. Ribosomal RNAs are transcribed from ribosomal genes. Here are mitochondria shown in red. Here is a vacuole. Here is the Golgi apparatus shown in purple, indicating that in purple. There's the Golgi. Let's look at the endoplasmic reticulum here, shown in kind of blue. You can see ribosomes are studying this. That would be rough ER. Here are some intermediate filaments shown in yellow. And if we look at the microtubule organizing center, that would be right here, and that would consist of centrioles that are organizing these microtubules shown in orange. Note the extensive extracellular matrix on the outside of the cell. Now let's look at some movies of cells and look at some of the dynamic features of cells as shown by a variety of techniques in microscopy. This video is showing the ruffling of a membrane, and this is called a cytoplast. It's an extension of the main cell, and so it doesn't have a nucleus. It's just connected by a thin cytoplasmic bridge. But notice the extensive ruffling of the membrane. This is caused by polymerization and depolymerization of actin microfilaments. And notice the main cell body is also undergoing this ruffling motion. And many cells use this ruffling as they migrate across a substratum. So again, here's an actin-based process, microfilament-based process. This video is showing the formation of large dorsal ruffles in a cell that's been treated with a growth factor. So time 0, it's hit with growth factor, and then you see these rufflings, which are actin-based ruffles that form. You can see other actin microfilaments in this cell. All the actin microfilaments are stained green because monomers of actin have been coupled with a green fluorescent protein. And so we're tagging actin that way. And you can see that these ruffles coalesce into a large heart-shaped structure, although the shape of that is not constant. That's just an artifact of this particular cell. So this is a cell responding to a protein growth factor by forming these ruffles, which are actin-based structures that help internalize components from the cell surface. In this video, we're seeing the polymerization and depolymerization of microtubules shown in green, and actin microfilaments shown in red. You can see a high concentration at the cell periphery, but also more internally in the cell. And you can get an appreciation for the dynamic nature of these cytoskeletal elements as they are polymerized into long fibers and then shortened into shorter fibers and then repolymerized into long fibers. There's quite a dynamic interaction going on. This is a human cell with the DNA stained blue. In this case, the microtubules made out of tubulin are stained green, and the actin microfilaments are stained red. And these cells are being treated with an inhibitor of microtubule polymerization called nitazoxanide. And you can see the disintegration of the microtubular network. Here it's intact, here it's disintegrating, but the actin microfilaments remain intact. The red microfilaments remain intact, whereas the green microtubular network is affected by the inhibitor of tubulin polymerization. And this is a nice example of how drugs can be used to study the cytoskeleton. This video is showing actin in a purplish magenta, so we can see actin microfilaments causing ruffling at the cell surface, at the periphery of the cell. And in green, we have a green fluorescent protein tagged myosin protein called myosin 2B. And remember, we talked about myosin being a protein that could associate with actin filaments and move those actin filaments by grabbing them and causing them to slide. And so we see both proteins visualized in this very dynamic process here at the cell periphery. In this video, taken by researchers at the National Institutes of Health, we see a nucleus of a cell, and we see fluorescently marked the endoplasmic reticulum. It's a complex network of canals in the cell. And now they're going to animate this with computer animation. Here's the mitochondrion. Here's the nucleus. Here are nuclear pores. And as the mitochondria kind of floats around the corner here, we're going to see that the endoplasmic reticulum is shown in blue and the rough ER in kind of gold. And the Golgi will be in green, as we will see. And what we're going to watch first is part of a vesicle budding off from the endoplasmic reticulum and finding its way to the green Golgi. So here we have some rough ER, and we're going to watch this as a piece of this buds off. A vesicle, oblong vesicle breaks off, carrying protein contents that have been synthesized there and fuses with the Golgi. That was the cis phase. And now in the trans phase, we see vesicles budding off from the green Golgi. Here's a close-up of that. And the surface of this is studded with other proteins that mark it for destinations in the cell. So this is a nice computer graphics animation of the inside of a cell. Simplified, of course, but nevertheless very beautiful and informative in its way. And then we go back out, and here is the view of the dynamic endoplasmic reticulum in the cell. It's quite a complex network of canals through the cell. And I'll just leave it up here so you get the credits. This is a beautiful animation by BioVisions at Harvard University. This is by BioVisions. And we're going to start off with a view of a small capillary containing blood cells flowing, and these are neutrophil cells rolling along the endothelium of the vascular capillary. This is a view of the membrane of the cell, and these are lipid rafts floating on the surface. These are interacting molecules here on the cell surface, and this is the actin network. Here are actin filaments. You can see the double helical nature of the actin microfilament. And here we're seeing actin polymerized into long filaments. So the globular actin monomers are being polymerized into long filaments. And here we have proteins that come and can cleave actin microfilaments. Here we see alpha and beta tubulin assembling into long microtubules and now depolymerizing. And here is the motor protein kinesin walking along a microtubule carrying a vesicle with it. And kinesin actually has this three-dimensional shape and walks just like that. That is actually true to the molecular conformations of that protein. It looks like someone walking. Here is the nuclear pore, and messenger RNAs are emanating from the nucleus out through the nuclear pores. And here messenger RNAs are being read by ribosomes, and proteins are being produced as the ribosomes read the messenger RNAs. Here we have ribosomes translating messenger RNAs on the rough endoplasmic reticulum. Actually, these are ribosomes translating messengers there. And here we have kinesin again, walking along a microtubule, dragging a vesicle, membrane-coated vesicle with it. Here is the endoplasmic reticulum, and there's kinesin again. And now we see molecules that are being exported out of the cell to the extracellular space here. And here's the extracellular matrix. And proteins on the cell surface can interact with other proteins on other cell surfaces. And that's what was going on when this neutrophil was rolling along the endothelial cells of the capillary. We're seeing that now. So we peered inside that endothelial, or that neutrophil cell. Now we're going to spend a few parts of lectures talking about membrane systems in great detail. We'll examine structure and function of biological membranes.